G'day, welcome to Navadia. Today I wanted to talk a little bit about the science of DNA, how we amplify DNA samples, why we do it, and more specifically in regards to a process called PCR or polymerase chain reaction. The reason why I wanted to do this video is that I've seen a lot of things pop up in regards to the science around coronavirus, and I thought I'd explain a couple of things for people who might not understand what PCR is. By the way, the YouTube gods have demanded that I bend the knee, so please remember to like, share and subscribe this video. Yes, I have now officially sold my soul to the algorithm overlords. Why do we do it? Amplifying DNA, I mean, not selling your soul to YouTube. There's a lot of reasons why we need to amplify DNA samples. For example, only 1% of microbes grow in artificial cultures such as petri dishes. We know this because after using other identification processes such as PCR, scientists were able to find DNA sequence that didn't actually belong to the microbes that were found growing in the lab cultures. Since viruses don't grow without a cell host, PCR is used to identify and sequence their DNA or RNA, depending on the type of virus. Another reason to do it is that scientists may only have a tiny sample of DNA, such as hair or a bit of skin, and they need more of it for forensic purposes. It also helps in archaeology. A bone from an ancestor that's 80,000 years old contains DNA, but you don't exactly want to destroy the whole bone, so PCR is used. These are just some of many applications for the amplification of DNA. Before I get into the nitty-gritty of PCR, I will give you a quick rundown on the structure of DNA. We've all seen the image of DNA as a ladder that is twisted into a double helix structure. I would also hazard a guess that most of you would know that there are 4 base pairs: guanine, adenine, thymine, and cytosine. And if you can even think further back to high school science, you might even remember that guanine and cytosine pair, and adenine and thymine pair. Some of you might even know there's even a 5th base called uracil, which tells the cell's machinery that it's looking at an RNA molecule by swapping itself out with thymine, which I think's pretty bloody cool. Fun fact, DNA stands for deoxyribonucleic acid. Another fun fact is I've known how to say that since I was about 11 years old. Thymine, cytosine, and uracil are known as pyrimidines, and they are called this because their molecule has a pyrimidine molecular structure. A pyrimidine is a 6-sided carbon ring, but the 1st and 3rd carbons are replaced with a nitrogen. Adenine and guanine are called purines, which are named so due to the fact they have a purine molecular structure. This is quite similar to pyrimidines, but it has an imidazole ring attached to it, which is a 5-sided ring, where the nitrogens swap out the carbons at points 1 and 3 here. Since pyrimidines and purines are different sizes, they pair up together in the DNA molecule, which keeps the DNA structure a consistent width, ignoring any mutations. These base pairs are bonded through hydrogen bonds. The reason why C only pairs with G and T only pairs with A is because C and G have 3 points where they have hydrogen bonding, while T and A only have 2. Hydrogen bonds are also quite weak, so please keep that in mind for the future. This might seem like a bit of a tangent, but I can assure you it is not. I want to introduce you to a groovy little microbe called Thermus aquaticus, which is found in hot springs in Yellowstone National Park. It's gram-negative and lives in temperatures of 70 to 75 degrees Celsius and at a slightly alkaline pH of 7.5 to 8. The reason why I bring this up is because these microbes produce an enzyme called Taq polymerase. Because this enzyme structure is stable at high temperature, it allows the bacteria to replicate its DNAs at temperatures that would normally kill other microbes. This enzyme is used in the elongation phase of PCR. Don't worry, that bit will be a bit clearer further on. Let's say you had a sequence of DNA you wanted to amplify. You start by identifying the sequence you want and choosing the primers you need. Without going into too much detail, the primers basically tell the Taq polymerase where to start copying from. Choosing the wrong primers means that you amplify the wrong segment of DNA. You have your DNA, you have your correct primers, so what else do you need? Well, you need nucleotides. Remember guanine, adenine, thymine, and cytosine? They're called nucleotides. These are just hanging around in the solution, minding their own business, doing their own thing, for now. The next thing you need is the Taq polymerase. I talked about this earlier. Taq polymerase identifies in the single-stranded DNA which nucleotide it needs to make the complementary strand of DNA and gets the correct nucleotide that is running around free in the solution. So basically, it turns a single-stranded DNA molecule into a double-stranded DNA molecule. Remember when I said hydrogen bonds were weak? Well, this is where I'm finally going to start talking about PCR. To break a bond in chemistry, you need to put energy into it. That could be either light or heat energy, even though technically they're the same thing. Since hydrogen bonds are weak, they are more prone to breaking than, say, a triple bonded carbon. In the first step of PCR, the solution of nucleotides, the DNA you want amplified, the primers, and the Taq polymerase gets heated up to the point where the hydrogen bonds break in the DNA, denaturing it. This is roughly around 94 degrees Celsius. It then gets cooled down to around 55 degrees Celsius, so the primers, which tell the Taq polymerase where to start coding from, anneal to the single-stranded DNA. The mixture is then heated back up to 72 degrees Celsius, so the Taq starts working its magic by reading the DNA and telling those freeloading nucleotides to do their work and become a bloody DNA molecule. The lazy bastards. The DNA helix then forms again, so the whole process of heating it back up to 94 degrees Celsius, cooling down to 55, heating again to 72 degrees Celsius, and then heating up further again to 94 degrees happens over and over again until you get the amount of DNA you need. From one single strand of DNA, you can get a lot of it within the space of an hour. These temperatures and times may change from machine to machine or reference material to reference material, but they are all roughly in the same ballpark. The calculation for how many DNA strands are created is 2 to the n, where n is the number of cycles, so it is greatly exponential. The time it takes depends on how long the DNA sequence you want to be amplified, but the first 2 steps normally take around a minute each. Fun fact, Taq polymerase at optimal conditions can get through 1,000 base pairs per minute. Wow! Usually 30 minutes to an hour of PCR cycles should be enough to get the amount of DNA that you need. This is all well and good, but what do we do after getting all the extra DNA? Excellent question. Do you recognize these things? They are the results of something called Agarose Gel Electrophoresis. You see, DNA is a slightly negatively charged molecule. We use that fact to our advantage to separate the DNA strands we made through PCR according to molecular size. I'll explain this later. But keep in mind that this is only for linear DNA, which is not circular DNA, which is found in plasmids in microbes. Also keep in mind, gel electrophoresis isn't the only thing they do with the amplified DNA. Other things they might use is mass spectroscopy to figure out how heavy the amplified DNA molecule is. To do gel electrophoresis, you make a gel which is usually, but not always, agarose. It is heated in a buffer solution such as tris acetate until the agarose is melted. Microwaving this for a few seconds helps this along. Before the gel sets, a highly toxic chemical called ethidium bromide is added to help the DNA to fluoresce under UV light. The gel is put into a tray and a comb is added to make the wells. Once the gel is solidified, the comb is removed. The same buffer used in the gel is poured over it until it gets covered, and that's usually by about 1 millimeter or a bit more. The DNA from PCR is mixed with chemicals such as Phycol 400, which increases the product's density so it will sink into the wells. Another product they use is Orange G, which colours the samples so you can visually see the progress of electrophoresis. These aren't the only chemicals they'll use for these reasons, but they're just an example. An electrical current is passed through the electrophoresis setup, which causes the DNA to migrate towards the positive end. As I mentioned earlier, DNA has a negative charge. As we all know, opposites attract, and that is why DNA is pulled towards the cathode or positive electrode in the electrophoresis setup. This also takes about 30 to 45 minutes to complete. From starting PCR to finishing electrophoresis is approximately 3 hours, give or take, depending on the length of the DNA sample that you're using the PCR for. The bigger the DNA strand, the slower it moves in the gel. This is because there's more resistance, kind of similar to wind resistance. Smaller DNA strands pass through the gel faster. A sample known as a ladder is also added to the first well. This contains a mixture of known lengths of DNA to compare to the unknown lengths in the sample. In the case of forensic science, the DNA of a suspect is also added to the wells and are compared to the results of the unknown criminal. Mutations are also picked up on because different mutations will cut the DNA at different lengths. The difference between gel electrophoresis and mass spectroscopy is that electrophoresis looks at the length of a DNA sequence, where mass spectroscopy looks at weight. If you have a DNA sample that has a lot of T and A in it, the weight would be different for a sample with lots of G and C, even if it's the same length. As you can see, PCR and the following technique of gel electrophoresis is a powerful tool in many ways. This technique is what they use for testing potential COVID-19 patients and is used to track mutations in the disease. It was also most likely used to debunk the myth that it was bioengineered. It isn't. It has too many mutations from the virus found in bats and pangolins, which is where they think the virus originated. If COVID-19 was engineered, there would be a lot less mutations. Outside of this pandemic, PCR is also used to diagnose genetic disorders, see if a gene introduced to a GMO has been taken up by the organisms, species identifications, and much, much more. It is a really powerful tool. I hope you enjoyed this and learnt a little bit about how we test DNA, some of the uses of PCR and gel electrophoresis, and frankly, how awesome science is. Links to my sources are of course in the description below. I hope you all have a wonderful day, evening, night, whatever, and scandium, iodine, nitrogen, and cerium. I also want to say a massive thank you to my wonderful patrons, especially my $10 Redback Spider patrons, Aided Furball, Lauren Hart, Amanda Vogue, Mary Civitano, Ross Devereux, and Atheist Pastor. I also want to say a massive thank you to my $35 Irakanji patron, Osman Cumming. Stay safe everyone, and please look after yourself and others. All the best.